Hello, my name is Eugenio Flores, and welcome to another short lecture in the Honors Chemistry Short Lecture Series. In today's short lecture, you will be learning about what is the gut microbiome, the effects, and the influence it has in our bodies. The second topic you will be learning about is an important analyzation tool that scientists used called the mass spectrometer. And lastly, and the third topic, we'll be learning about the gut microbiome and the effects it has on the pills that we take. Now, on to the presentation.
Specifically, in this presentation, we will be learning about what is the gut microbiome, what does the gut microbiome do, what is mass spectrometry, why is mass spectrometry used. We will be learning about two drugs that are popular and shows the reactions it has with the gut microbiome and how it is affected by it. And lastly, the future of probiotics and regulations. Now, let's get to the most important question in this presentation. What is the gut microbiome? The gut microbiome is located in the gastrointestinal tract, which, in other words, is called the digestive system as we know it. This gut microbiome is made out of microbes, which is an organism or a microscopic organism that includes bacteria, fungi, archaea, and protists. However, things like viruses and prions are not considered microscopic life because they are not alive in the first place, so they're not considered microbes. The gut microbiome contains enzymes, which are super important for metabolism, breaking down our foods, and absorbing chemicals, which is more prevalent, as we know, in our medicines. As you can see in the picture of the right, most of the gut microbiome or microbes are in the large intestine, which is where most of our food, pills, and chemicals go through. Next, we will learn about the functions and effects of the gut microbiome. As we know, the gut microbiome is part of the digestive system, which plays a big role in our bodies. One of these roles is that it's an immune system support. It supports the immune system by alerting that there are pathogens, diseases, or bad chemicals in our intestines or digestive system. And by doing this, is able to protect the body. Next, it has a good communication with the nervous system. By controlling the amount of blood that is circulated in the intestine, the brain is able to send communication to our intestines to show if anything is wrong or needs to be taken out. Next, and the most important one in this presentation, is the role of metabolism. It metabolizes many foods, medicines, and chemicals, which is important when we're eating food, taking pills, and putting chemicals in our body. And as we know before, the gut flora, which is another name for the large intestine, most of the microbes, bacteria specifically, is located in the gut flora. Before going into the reactions in a gut microbiome and pills, we must first learn what scientists use as a tool to analyze and record the data. These scientists use a mass spectrometer. This device was extremely essential to projects that have to do with analyzing specific changes in a molecule. This helps scientists not only find specific changes in a molecule, but also identify unknown substance or substances that barely arrived into the molecule and changed it. Now, there are three essential parts to the machine. The ion source is a process in which the molecule converted into an ion, so it can be manipulated easier. As you can see on the chart on the right, these are molecules, and by making them positively charged, into a cation, they're able to be manipulated easier on this area and able to be consistent throughout and record in the mass analyzer, which is used to weigh every single one of the cation to record changes in the molecule. The detector, which is this, is able to not only filter out which ions are different, but able to detect new substances that the molecule has. These changes help scientists very much in discovering what happens to the molecule itself and what changes that happened before. Let's move on onto the main topic of this presentation, the CNN article. This article talks about reactions between the gut microbiome and certain drugs. The two drugs I am explaining today are the very popular irinotecan and the levodopa. However, we're going to talk about the irinotecan first. Irinotecan is a drug used for colon and rectum cancer. It is a prodrug, which means it is deactivated at the first place and is a molecule. What it does is that it travels to the cancer site and stops the cancer cells from multiplying. It completely stops. It also affects other parts of the body too and causes some damage, like diarrhea, sometimes it clots itself, and it's typically a very risky drug to take. However, this has been considered by scientists to be a phenomenon that doesn't make any sense, since human enzymes are not the ones who are causing this. Now, let's go over the route that irinotecan is supposed to be taking. Once you swallow irinotecan, it's supposed to travel down the intestine and get absorbed by the bloodstream. To the bloodstream, it goes to the liver, and liver enzymes compound it and combine it with the sugar compound, which deactivates it. What deactivate means is that it doesn't cause any damage to the body. It's technically just a molecule floating around in the bloodstream. However, it has been recorded that once the deactivated irinotecan reaches the cancer site, it activates and it does what it has to do. However, the stragglers, or the ones that do not make it in the cancer site, travel down the intestine. It is shown that it is activated in the intestine, which causes damage. Now, why is that? Apparently, once the deactivated irinotecan enters the intestines, there is a weird phenomenon in which the deactivated irinotecan suddenly becomes activated. Scientists then studied and tried to figure out why this happened. As you can see the chart on the right, this is supposed to be the formula. Activated irinotecan is added by a sugar adding enzyme, and technically it becomes a deactivated irinotecan. The sugar acts like a kind of a shield or something that kind of paralyzes the irinotecan from doing anything else. However, scientists have figured out that there are bacteria that eat or identify the sugar as energy and take the sugar away from the irinotecan. After the bacterial enzyme takes away the sugar, the irinotecan becomes activated once again. And it's not good news when an activated irinotecan is activated inside an intestine, because that means it's activated and can cause damage to the intestine, which causes gastrointestinal side effects. Not only does this include diarrhea, stomach aches, and a bad stomach, it can also cause very severe cases of people being in bed for a while and not being able to move at all. And sadly, this medicine has been really bad for people who have these bacteria inside of them. Another drug that greatly reacts to the gut microbiome is levodopa. Levodopa, as you can see by this formula right here, is a precursor for dopamine and is used for Parkinson's disease. Parkinson's disease is an illness in which a person's brain cannot produce dopamine. So they see levodopa as a safe means of getting dopamine into their brains and curing their Parkinson's. However, you might be asking to yourself, why can't dopamine just be taken and the people with Parkinson's don't have to deal with levodopa? It's because dopamine cannot cross blood to brain, since it can damage the body and it's considered an active phase drug, which you don't want to swallow an active phase drug if it's going to damage you in the first place. So levodopa is used. Now, scientists have been debating in which levodopa is strong enough for it to get to the brain safely. And they have considered putting the carboxyl inhibitors to prevent losing that one carbon atom and losing its phase as levodopa and to dopamine. However, that has not been consistent enough, and scientists were confused on why that was, since human enzymes were not the ones breaking down this. Now, as I've stated before, scientists thought that a deactivated levodopa was enough to get the active phase dopamine into the brain safely. However, scientists have pinpointed two species of bacteria that have not only put dopamine outside the brain and into the bloodstream, but also create a harmful substance named amphetamine. The first bacteria was the Enterococcus, which makes levodopa into dopamine by making it lose one carbon and two oxygens. By doing this, it's already damaging the body by putting the dopamine outside the brain and into the bloodstream. The second one is even worse, which is Agarthella, which makes dopamine lose one hydroxide, meaning that the dopamine turns into amphetamine, which is an extremely harmful chemical in our bodies. This is an active phase amphetamine, which means that it negatively affects the body. By doing this, amphetamine is causing bad diarrhea, bad stomach aches, and can put people in a bed for a while. With increased cases of drugs like levodopa and irinotecan being found out every single day, there was a study done by Andrew Goodman with a team of Yale Medical School students. They tested over 271 popular molecular drugs such as Gleevec on 76 dominant gut bacteria. And to the researchers' surprise, 2/3 of the drug were metabolized by the bacteria, which is a huge amount of drugs considering that many people do not know, or many scientists do not know, that these gut bacteria are metabolizing these medicines. Also, most drugs were affected by almost all of the bacteria, and some were not at all. This should be a warning to future companies to not only calculate the gut bacteria and put into equation what can happen if they don't account for it, but this can also cause many people to not only get harmful side effects, but also die from those side effects. However, few companies have been looking towards probiotics, which is another way of counteracting these gut microbiomes and the microbes that are there, the harmful ones that are causing the metabolism of unhealthy chemicals in our body. In conclusion, we have learned that the gut microbiome is an essential part of the process of activating and deactivating drugs, that many companies must take into account the importance of having medicines that are immune or are less affected by the gut microbiome, and by manipulating the gut microbiome, companies will find it easier in the long run to release medicine that either works well with the microbes or ignore it completely. Thank you for taking your time to hear my presentation. Thank you.